Yes.
Ja, nou het is niet helemaal anders, maar het is voor de verschillende berekeningen goed natuurlijk. Ja, maar dat moet je niet wel doen. Ja, anders is misschien wel belangrijk, is jullie
Nee, maar dat is 5,2.
Oh, oké. Ja.
Kan ik niet zelf lopen dat automatisch?
Ja, dat doen wij. We hebben nog niet heel ingevoerd nu. Dan gaan we dat vergeten en dit is even aan weer. Ja, en dan sturen we naar nee, sturen we mensen op. Ja, dat is mensen kunnen op de trap zitten. Good morning, everybody. A few small announcements. Keep the lights on. Okay. So, first of all, I apologize for the room. It's too small. It fits 100 people. We're with roughly 120. We're trying to get bigger rooms. There's a couple of seats left over here, over there. So please shift into the empty seat. We're trying to get bigger rooms for the coming weeks. For this week, the whole campus is full. That's one. Second thing is, if you haven't completed, who has not completed the survey on Canvas for the background and practical methods? Please do that now. Or in a break, do it in a break. But because we need that information to be able to sort you into groups properly. Right? If not, we'll make a guess and we'll make a mess, maybe. Yeah?
Is there a few questions as well from 30 years then? But I think that's an error.
Yeah, probably. That's fine. Yeah. Sorry for that. Okay.
So then you don't need to fill out number 13, right?
Then yes, and the third thing is, if you have time, you don't have to do it today, but please put a photo on your profile that helps us to start recognize your faces and put a name to it. That makes things a bit easier.
It doesn't only help us, it also helps you.
Yeah. It helps you to help us to help
friend
you.
one another.
Yeah. Okay, I think that's it. Oh yes, stuff is recorded. Yesterday's recordings went fine. They should be on Canvas as soon as they finish processing, which took a long time. We started last night, and finished this morning. But they will be online at some point in the future.
Having the camera there is probably better when I move over here.
No, that's fine.
No, I stand in the way of the screen, I guess.
Oh, yeah, but you can look at the slides. You're more important.
Really?
Yeah. Okay. Somebody please remind me next time. I should put the video on top of the beamer. It's a very convenient spot. The beamer is noisy. Okay. There's one or two chairs. If you're sitting next to an empty chair, please help the people that come in to find it. If your bag is on a seat, that means it's empty. I'll see you guys at 11, and although I think we need to keep the door open because of the air.
It's just a little bit too windy. Yeah.
Well, they will arrange it really becomes terrible.
You can try.
Close that one, because that's the whole way.
You can try the difference, but it's...
No, we'll close this one.
Yeah. It's yours. Is there anybody sitting next to an empty seat still? So hopefully next week we'll have bigger reviews.
That was the start of the lecture. Let's sound. Now let's see if first if you open this one again. So, welcome once again. Ooh. I cannot guarantee that this door will not slam again, but okay.
Maybe a trash bin. Maybe a trash bin somewhere.
Yeah, well, of course. There's a table over there. Seems fine now. Second day of your master program. So tonight you'll be here twice as long as yesterday. Isn't that amazing? Right? I'll probably switch things off. Are you going to fall asleep at this sort of intensity, or will you be fine, you think? Okay, good. Right, so I'm going to fill you in a little bit on genes and proteins. Maybe most of this stuff would go more in the so-called biology conversion class, but let's, we'll put some in here. I think I showed this slide last Friday, for those who were there, during the, should put this in here somewhere, where you see DNA. And so DNA is a molecule. You see a little bit of the DNA structure here. It's the so-called famous double helix. And who knows the Watson-Crick base pairs already now? And who is totally flabbergasted even hearing what I say now? Right? Well, it's an awesome molecule, DNA. I'll show you that later on a special slide. But there are basically only 4 bases. And they're encoded with the 4 letters A, T, C, or G. You already see here. Hey, I'm lying. You see N's, but N means it's a result of measuring a DNA molecule, finding out all of the bases that are in there, that sometimes the machines are not helpful enough, and people get out of the data that there is something here, there is a base, but we don't know which one. So N is the generic letter of something. Yeah, so we know the length of the molecule if you do this, so it's good information. to sort of put in, there is something, but hey, we don't know what it is. One of the four, okay? Now, the beauty of this is Watson-based variants. You see here these little structures here, called that glue for now. These two strings of DNA are glued together by special so-called base pairing, and I'll mention it, but later on in your class you'll hear it. C sticks to G, so a C here will stick to a G there, and an A here will stick to a T there. So there are only two combinations possible to stick the two strings together out of the four letters. Yeah? And that means, by the way, and that's a nice sort of side effect of that, the system knows all this, right? It operates according to those rules. We only have sort of found out about these rules that biology gives us. But if you have one string like this, you can just immediately fill in the other half. Shall we start doing this? What will be the other one here? T, G, G. Good guys, you get the hang of it. The only thing you need to remember is that if this string here runs in this direction, DNA has a direction, it really sort of starts here and ends there, but then the other string will run in the other way, right? So this is what you have with DNA. The one is encoded in this direction, so this follows that, and the other string will have, the other side following the earlier half, okay? So, that's one thing. So I'm going to explain to you how you get in the end from the DNA structure, the DNA sequence, via genes, and the genes is sort of a localized little piece of DNA. Well, localized, it can be 2 million long. The longest human gene is roughly 2 million letters, but the short gene might be a couple of hundred letters on the DNA. How that is encoded into a protein, and here you see a nice picture of the three-dimensional structure of a protein. Now, DNA basically is a dull structure. Three-dimensionally, it looks pretty much like this. Not totally, sometimes you have cruciform structures, so you get sort of a cross here and there, but the ballpark figure over the whole, is mostly like this. Proteins are more crazy stuff. They fold up in all kinds of weird three-dimensional shape, Alzheimer, I'm already becoming afraid of it, you have time, right? Anyway, what happens? You have a structure, and normally the molecule is like this, and now at once it develops two Mickey Mouse ears. Two structures, and because of that, more Mickey Mouses come in with those ears, and they nicely stick together. So you get, and the problem is with this disease, you get an incorrectly folded protein, so with two sort of ears sticking out. Other one think, oh, that feels good, I'll do this as well. So proteins go wrong, the structures are disrupted, and then you get an enormous protein plaque, so much in an innocent brain cell that the cell thinks with, this rock inside, I'll give up, I'll die. And that's a very good process. Cells die many times, but it should be for the proper reason, not for reason like this. And that means you really get little holes in your brain, and where there is a hole, there is no thinking. Or remembering, and that's what you find out that people will suffer. So basically, the protein structure is terribly important for what the stuff is doing, okay? If we would understand, and I'm going to look at this a bit more, if we would understand, given a protein sequence, talk a bit more on that later on in this lecture, if we would understand, given, the system knows it. If you have a gene for a hemoglobin sequence, it folds normally like a hemoglobin. So the system knows. So we can't get our hands behind it. What are the rules? Sometimes you feel, what are the bloody rules behind it? We should find out. Folding can be very fast. It can be a split second to do it. So bigger proteins, they need to think more. What should put this on there again. But, so there should be some secret, right? And it's probably physics. But we start getting some. Well, we don't get anything maybe, but I tell you what we do is bioinformatics. You're here because you do a cheap trick, basically. But I'll fill you in on that later. Okay, so what you already can see here from DNA, we go via another thing that's called RNA, and you can already see a difference for those of you who are quick at recognizing letters. What do you see? Look in in blue or no, already in red. No, here. You see this letter? That's another one, right? It's not these four you find here. So that means that a T thymine is becoming a uracil for the encoding is still 4 letters, but 1 letter, you know, just for the sake of it, you know, I'm RNA, not DNA, so I want to look a bit different, right? So I'll stick in a U instead of a T, okay? This is human reasoning, this is not how biology reasons. But anyway, you already get the hang that sort of 3 letters at the DNA level or RNA level, that's the same, have something to do with encoding the letters in the protein. See this picture that even the color scheme. Yeah, so, okay, but I'll get back to that later. Okay, so you see a 2-step process here, that's important. And what that means is this first step is called transcription, to convert a huge DNA molecule, a localized bit of that, into an RNA structure, or sequence if you like, which normally is a lot shorter, of course, than the DNA, right? How long is our DNA? Who knows? 2 million?
No, 2 meters.
2 meters? Ah, no, much more. Roughly. That's a very good point, by the way. How many base pairs or letters do you think there are in human DNA?
10 to the power of 9.
Yeah, roughly correct. You're ballpark figure, yes. So 3 billion a bit, 3.2, 3.3 billion, right? And then you would instantaneously think, but we are, of course, the best created creature on the planet, so that must be the biggest DNA, was so great. So a water lily laughs at us 30 times larger, a salamander laughs even more. So there are vast number of creatures that have much more, much longer, or you know, more base pair containing DNA. And then there was this other secret that thought, okay, if we won't win here, we should win onto something else. You know how we could win still. We have more genes than all the other animals, right? So we start counting genes. Deception again. Yeah? We're not any great. We're just mediocre on all accounts when we look at it genetically. You know? Sorry, visit the shrink if this is shocking to you, but you know, this is just what is happening. Anyway, more on that later. So, transcription and then the translation process, why is it called translation? Because we really are going to translate a set of 4 letters into a set, if you like, of 20 different letters. There are 20 different bases called amino acids to build a protein. Yeah? So we have the amino acid P and E, and these letters mean something, right? This one is, for example, called proline. You know, so it starts with a P, so it seems to make sense. But this one is a glutamine. So you have to give out 20 letters to some molecules. So, but you know, by the way, when I started studying, they said, and I'm not going to do this to you, so be happy. Tomorrow, you know all the 20 amino acids by heart, the structure, and of course their full names, right? We had to memorize in those days. Because your brain is, you know, you have Google, so you doesn't need to be here anymore. You know, just look at that, right? So that's fine too, it seems. So anyway, I'll do this thing. I showed you this picture last Friday, doing an inline poster. So this is sort of the complexity of get metabolic pathways, but there is a lot more to entertain in a body than metabolism alone. And this is one of the important networks that we have. Now, go quickly back to this slide. So how do we get, by the way, how is that encoding done to get from DNA to a whole bunch of proteins? Yeah, there are roughly 20,000 genes. So there should be roughly 20,000 different types of proteins, different sequences that will probably fold up, well, most of the time, in different structures. So how do we get to that complexity? So here we go. Now this is DNA. Now we take a little bit more structural view of DNA. Well, it's simplified, of course. Now it's just one line on a sheet of paper, if you like. But here you see an important little thing. There's just a little bit of DNA there, maybe 20 bases long, roughly. And this at once we call, hey, this is important. This will encode something. Basically, DNA is an information-carrying molecule. That's very important. It has a structure, it's doing physics, it's doing all of that. But it's used to sustain life by being, converting information, containing information. That is important. Okay, so, and one aspect of that information is a so-called transcription factor binding site. It's a great title, you know. The transcription factor is a protein, and then what is it doing? It binds to something, and how is that called? The transcription factor binding site. So the name of the thing tells exactly what it is. That's great, right? That's clever naming. And there is quite a bit of clever naming in biology. It's an awful lot of stupid naming, but anyway, another piece of clever naming is protein names. Protein names also very often carry what they do in their name. Amylase, ACE means it's an enzyme, and amylase means it does something to amylase, and that's a sugary type molecule. Alcohol dehydrogenase, you get the hang of it. It's having to do something with a substance you might take every now and then. Anyway, let's talk about how to make a gene. We're here now at the transcription level. How do we get from a DNA molecule to a gene, to an messenger RNA molecule that is sort of a copy of a local piece, of a local substring, if you like, of DNA, okay? So normally that is, in biology books, when the gene starts, it's given by this sort of thing here, it starts and then it's an arrow. Why this arrow? That makes sense. Basically, Paul Toos, this is of course geek speak, right? Biologists know immediately what it is. This is polymerase type 2, that's the type of big protein complex even, that's actually bigger than that guy, here in the schematic drawing, that will act as a copying machine. This molecule, is a point of attachment. Also, this is a motif carrying information, Tata, baby language, which I'll get to you after my line, that allows the Paul to attach to the DNA if this thing is not there. If this is mutated away, sorry guys, this polymerase is not going to bind. So conservation of properties is important, but when it does, it will start and then copy that local bit of DNA until DNA tells it, now you can stop. I'll get back to that later. Yeah? So this molecule arranges a local copy, converting DNA, a local bit of DNA, into a messenger RNA molecule, which eventually will lead to a protein. The proteins are important for us. Proteins are the doers, if you like, in the body. Okay? Now, but you don't want this copying machine go crazy, right? On a real copying machine, normally there's also a button you press and then it starts copying. You don't want the copying machine to always copy when it likes. Also in this system, and what is the on or off switch? That is being arranged by another type of protein called transcription factor. So this is a factor that has something to do with transcription, very good name. And how does it work physically? I'll give you a little bit of the hang of that. So this is a special molecule because it binds to a specific motif, a specific set of letters of DNA that allows this specific transcription factor, and maybe there are hundreds of different kinds of those in your body. So one will bind to this specific one, and that one will bind then to this polymerase. And what happens, in fact, is some physics happen because it binds here and binds there. This structure will change a little bit. Proteins are not very stable. You push them a little and they start deforming, if you like. And as a result, the polymerase changes its structure such that it will start sliding over the gene and copying the DNA into RNA. So it just gets a switch, if you like. Yeah? So it is, and now the beauty is, so you have all kinds of little bits of motivic information here, and if there would be a different bit, if here there would be a mutation, this transcription factor might not bind anymore. It might be that another one binds, but if nothing binds anymore, what happens? This gene is silenced, as you call it. Cannot be formed anymore because the messenger RNA encoding that gene will not be produced. Yeah? Okay, so this is sort of thing, and then there is a lot of stuff. You know, DNA has to band, and DNA is pretty stable, double helix, so you can imagine you cannot just do this, the physics won't allow, so you need to take it a little bit more of a curve. So there is a minimum number of base pairs that should be here in order to bend it, right? A big rod of metal, if you want to do this, is hard, but you know, maybe if you do it like this, it's easier. So that's also happening. But importantly, all of these properties together allow DNA to tell a story, to convey information. Now, textbook pictures of all of this. This is the bacterial situation where normally you have a single transcription factor, you know, switching the polymerase, the copying machine, to produce the messenger RNA on or off. And then when we go a bit further in evolution to eukaryotic cells, I think Anton showed you the structure of those cells in yesterday's lecture. Then you see that multiple transcription factors together are a sort of more complex switching on or off machine. Okay? So you can do more. More combinations are possible. You know what this system wants to do? It wants to say, look, those 20 different genes I'd like to produce now. You know what? Because we need a big complex, but all of these proteins need to play together. So I should have all of them. So how do you create a system that flexibly can switch off or on, these 4 or these 3. And my question to you is, if you have time, think about how that might work given this system. And it does work given this system. I can see that for a fact because you all sit here happy enough. So that's very interesting. Also biology, right? But we're back again. Good. Okay, so this is the bioinformaticians look. This is these transcription factors you have here. Are for real for 2 sort of genes. GAL1 gene and the GAL10 gene. So here a messenger RNA needs to be produced from roughly here, and there a messenger RNA will for that gene be produced roughly there. And this is a nice example. I think this gene, if this gene is on the one strand of DNA, this gene will be on the other one, because of the directionality of DNA. The one strand runs like this, and then the other will run like that. Okay, and what do you see here? And this is interesting. These are 4 yeast genomes. 4 different yeasts. Baker's yeast, the one you know, I mentioned alcohol, and if you mentioned beer, then you have Baker's yeast, and bread, of course. But you see here that these are transcription factor binding sites. So you see these are indeed short little motifs. And in red you can see that what bits are totally conserved across those 4 species, right? This is the way when you put up those DNA sequences like this, you can look what is conserved, what stayed the same in evolution. I'll fill you in more on that more precisely later on. So these GAL4 transcription factor binding sites are important for the polymerase that will produce the messenger RNA for this one. Yeah? And MYC1, I think, is important for this gene, for that protein sequence that will be encoded by that gene sequence over there. Now, what you can see is already interesting, perhaps, that you would think, oh, one of those binding sites is enough, but here you find that for this gene, there are 4 GAL4 motifs. Now, I can talk as a scientist, it's a very pleasant job, it's a lot of anomaly, but you have to get used to disappointment, right? The moment you think, oh, I've established a firm principle, right? You wait a day and look again, and bloody hell, another species comes in, completely refutes your theory and so on. And then you're flexible, you start again. So here you can see this. People believe, we've seen a whole number of GAL4 motifs in different species, and you know what? We know for sure what the rule is. It starts with CGG, and then for 11 bases or so, we don't care so much. But then there is CCG. Fantastic, it worked for really quite a long time. And then these guys did this study, and they measured stuff, and bloody hell, this is another GAL4 motif, meaning what was established as totally true is not so much true anymore here. Because here it's not CGG, but it's CGC, which is different, right? So, not so conserved as people believe. Yeah. But then this is still there, the spacing is roughly okay and so on. Anyway. There are physical reasons why that is the case, okay? But informationally you can see, and why would you think, and I'll get to that later, and then I hope you'll find out, that's the reason why it is a good idea for a gene to have multiple binding sites to allow the transcription factor to bind there, and then kick the polymerase, the copying molecule, in motion, okay? Now, I told you, the system also wants to be able to express multiple proteins maybe at the same time, and then what you see is sort of gene networks where a whole ensemble of different transcription factors, each binding, acts together as an even more complex switching box. Saying, okay, these 4 now, and now these 6 later on, when there is another condition. Okay. So this is called the gene network, and we'll see many examples of networks. I showed you already a couple. This is not to be observed in your body, right? This is just molecules binding. And if it binds or does something, this is more one influence the other. How you read this is, a green arrow means if this gene is on, then the next step will be, it will activate, which will activate, for example, this one. CL will enable GATA. And the red arrows means if this guy is active, it will block that one, right? In biology, there's a lot of influencing in a positive or negative sense. So you don't see this really, so I'm going to show you the way how you can get to such an abstraction in a network like this, in a gene network. So here we go. What I just told you is this, right? We have a promoter, forget this now for the moment. This is the gene. The polymerase should bind somewhere here and be able to copy this gene over in messenger RNA. And here, upstream, there might be a little bit of stuff. These transcription factor binding sites are here because these blobs here are the transcription factors. They bind here. So this is a binding site. Here is a binding site very close to the gene. And also another thing that people didn't believe could be true was there are even binding sites that are in the middle of a gene. What does that mean? So this bit of DNA apparently is used as information to build a protein, and at the same time, it acts to bind, to allow a transcription factor to bind. But now, what happens, you think, if these transcription factors are here, bound to this bit, and now the copying machine comes and wants to slide over the DNA. What do you think will happen? It's like having the train and there is an obstruction on the tracks. It will stop maybe, or go bad, or whatever will happen. So that means, so what kind of influence will this one have on the making of this gene? An inhibitory effect, right? So that's physically, of course, clear. But what this thing is doing, we won't know whether it inhibits or will promote it, right? Anyway, so you see already complexity. Multiple transcription factor binding sites might make a single gene again. Now, these are called regulatory elements, these transcription factor binding sites. And now we can have such a situation again. So here you see the process going, here is the polymerase. This one is clicked into motion, and you see the messenger RNA molecule coming off the DNA. That really happens physically, right? This molecule, RNA, is produced for real when the polymerase reads the DNA. So this polymerase is a clever molecule. It should be able to read the base pairs, or the bases of the DNA, and then say, okay, oh, there was a T here. So in the messenger RNA, there should be a U, and it recruits the bases and ties them together in another string that is called messenger RNA. Okay? Right, so, there's different words for, where the place is. So proximal is there, and distal is there. So basically, it can be anywhere. Not, so now we're going to abstract it. If we have a situation like this, multiple genes, transcription factor binding sites for those genes, yeah? And now you can see, and these are all, by the way, the transcription factor is a protein. So where is that encoded, you think? A transcription factor needs a gene, as does any other protein need a gene. Do you see the loop coming up? Right? So, anyway, so these are just a bunch of transcription factors, but that means that this transcription factor might activate or inhibit the making of this gene. And here you see that a little bit already. You see, for example, that, how do you translate that? This one here, that blue one, inhibits the formation of gene B. So, transcription factor A inhibits the making of transcription factor B, and it will promote the making of gene C here. So an arrow is plus, and that thingy here, like here, means inhibition, minus sign if you like. Now, if we're now going to take this and abstract away from it, then we're at the picture I showed you a moment ago, where you say, okay, that means that gene A, look at it, what is it doing? Gene A is, this the blue one, is repressing B, is promoting C, arrow, and so on, right? From this picture, this is the physical world, we abstract away, make it into a gene network. Okay. Right, so, read this at home. These are all the bits and bytes you need to get gene transcription. You need transcription factors, which are essential. You need to have them bind to a DNA motif and so on, right? Okay, so this is something DNA does. That's very important. Now, DNA is so important as a molecule. It sustains our life. Without it, we couldn't live. Cell division is based upon DNA. It gets a very nice copy, a total high fidelity copy of any molecule when cell division takes place. So I would say it's at least as important as an electricity cable. How do we treat electricity cables? We treat them wisely, right? What do? We put insulation material around it. Why is that important? Well, to protect it and of course make sure that one cable is not sort of interfering too much with another one. That's another thing you get. But so DNA is doing something like that. And how does that look? That's called chromatin. But, with an electricity cable, the insulation material is around it to protect it, to stabilize. And DNA is a bit more crazy than that because it's not the insulation material. The material that stabilizes the DNA structure is sort of, the DNA packs around the stabilizing materials. So the electricity thread, winds around the insulation material. So that would not be a good idea for an electricity cable, I guess, but a good idea for DNA. Anyway, this is what happened. So DNA can be protected, right? Now start guessing. If you have this nice bit, so the DNA is wound like this round here, what do you think this poor transcription factor comes and it sniffs, here's my binding site. But then, bummer, it's packed up, I can't bind to it. So that's another thing that might happen. Although the information is there to be used, in practice it can't happen. So, to sort of abstract away, to show you this a little bit, these are these molecules that DNA wound around, nucleosomes, the insulation material if you like. And here is a very nice binding site, but we call it closed because the transcription factor cannot bind here. But it might be able to bind there. So do you see the cleverness of having multiple of these sites upstream? Because then, if this happens, still there is a higher chance that if these ones are blocked, at least that one, the pack, might be available, right? This is how evolution, how biology, how life develops, evolves, and sustains itself, okay? Right, so that is to give you a feel like how does information work, motifs that are there. Through evolutionary times, they might mutate away, and you get different systems, but during a lifetime, all of these processes, and this is overall, this whole field is called epigenetics, epigenomics. We have DNA, but this is stuff that happens, say, around DNA, but it interferes with the functionality of DNA. That is called epigenetics or epigenomics. Anyway, I'll leave it at that. But this is, you get a feel of some of the complexity that happens around transcription, making a messenger RNA sequence when DNA is available. Now, translation. I already told you, let's think a little bit about it. So we have 4 different letters. Now you're a computer scientist or an informationist, if you like. 4 letters in DNA. We need to build a protein where we have to fulfill the requirement that we should be able to encode 20 different bases. So we go from a 4-letter alphabet, DNA, to a 20-letter alphabet. Can we encode it 1-to-1? Is the information there? No. How many options do you have per site? Here 4. How many options should you have here? 20. So then biologists start thinking, say, brilliant, let's take 2 letters on the DNA. Is that enough? Do the combination, guys. 4 times 4, 16. I can tell you 16 is less than 20. Good. Not good enough. So what do we need to do? 3 positions. Do you remember that slide? That's 4 times 4 times 4. What's the number there? 64. And how many letters? 20. Ooh, now we have another problem, or not? What should we do now? Just say we use 20 and the rest forget about it. Would that be the way to go about it, do you think? Would that? That's another beautiful mechanism that's very important for evolution and for life. Redundancy. This sounds like a terrible thing, perhaps, right? Redundant is a negative word, but in biology it's beautiful. It just happens. Would I have done, I shouldn't mention this, but I will think for a moment about this bridge in Italy, but I shouldn't. But would there be more pillars? One could have broken down and the bridge would still be there. Right? If the bridge would be constructed, would have been constructed a bit more in a redundant fashion, overcapacity you could say, then safety increases, and also in biology. Oh, going back some reason. So here we go. Translation from a 4-letter alphabet to a 20-letter alphabet goes via codons, and codons are nothing else than just 3 letters in a row. That is used to encode an amino acid, a protein base. So we have 64 possibilities, and now we need to make some, we need to arrange this. Which sort of triplet codon at the DNA will encode what amino acid at the protein level? And now there's something beautiful. And that is a table, Rosetta sort of stone table you might say. Here you have the list of the 64 letter combinations. And remember, first you have transcription into messenger RNA. So really, the encoding uses the 3 letters on an RNA molecule. So you won't see T's, but you see U's, right? Doesn't matter, T would have been the same information. Here you see these little 3-letter codes here are just the 20 types of amino acids. But you see who has sharp eyes and see sharply. There is one sort of longer, funny, there are 3 funny amino acids. Do you see those? They're called stop. Now, I can tell you, stop is not an amino acid. Stop is the encoding thing for the polymerase, the train, to say, okay, you tell me to start here, tata box and so on, here is the first start. Where's the start? You know what, that's sort of hidden a little bit. There is one amino acid called MET here, AUG. This is the universal starting site. So the copying machine knows, I should start copying, but at what page should I start? Page 2, 3? You tell me, and here this is encoded by you start here. And where do you stop? Very cleverly at the stop codon. Yeah? So, if this is the idea, if always this copying machine starts at MET, what is by definition the first letter of a protein you think? That's not difficult to get, right? That's MET. It's methionine, okay? But then we look in protein databases, and very many proteins do not start with MET. You know what? And I will not talk any further. The first bit of the protein, you know, the system is so cleverly set, you know, it's amazing. Respect the cell. The protein need to go to some defined places in the cell. You go to the right, you go to the Golgi system, and you go to the endoplasmatic reticulum. I deliberately throw in a few difficult sounding biology words for those who need to get into this. So you need a postage, you know, there should be an address on the protein, you know. So the system, where do you want to go, right? And the protein says, look at my first 20 letters of the protein. This holds the information, labeling information where it should go. And then the system says, okay, then I'll read it, but then I'll take that off when I know where you go. And that is the reason. So the first bit of the protein normally is cleaved up. And then you do not necessarily have to see the methionine anymore. But the coding, I'm sure you started it. And then the stop, there are 3 different stop codons, okay? So this is the table. How redundant is this stuff? Do you see this? This leucine, for example. You see, this is one of the champions. There are 6 different codons out of the 64 that will encode this amino acid. Meaning, each of those will produce a leucine as a base. And what do you see? What about the 4th letter here? You see, when you have CU as the first 2 in a codon, the 4th letter doesn't matter what it is. All 4 will just produce a leucine. And if you look correctly, you see many of those examples where, you know, where the 4th letter here, all of these, the 4th letter, the 3rd letter in a codon is not met. So that gives DNA a chance. Now your DNA, right? And you say, look, I want to become stable and say there are some rules. Where could you start playing if you're inside a protein coding sequence? Because you have to adhere, if a certain protein should be produced, you can imagine that your DNA sequence is tied to this codon table, otherwise your protein will differ. So what mutation at DNA will not interfere with the protein? Typically one that happens in these 3rd positions. Because it doesn't matter. That's redundant, right? Okay, so to give you a feel of the redundancy, here you have the codons that will encode the leucine. You see that leucine indeed has 6 different codons, and the largest one of the pack, for example, called tryptophan. So this is the largest amino acid. There's only 1 codon, yeah, and also methionine because that's also a start codon. You know, so any mutation here or here will disrupt your system at the protein level. Okay? But not those. You can get away with a mutation as long, of course, if you land from this one in any of the other one in this list. Then the stuff remains the same. Yeah? If you hop over to another one, you get another amino acid in the protein. Yeah? Okay, so now, the polymerase was the copying machine to get from DNA to messenger RNA. And now we need to do the translation, the next step, from messenger RNA to protein. And that happens in a cellular thingy called the ribosome. The ribosome is a big blob of special RNA and proteins, ribosomal RNA. Anyway, this is a factory that really translates, because here is the messenger RNA coming in, and this is like a train copied into a 4-letter alphabet here into a 20-letter alphabet called a protein sequence. Okay, this is how it works in the cell. So this really physically happens. Okay, right. So DNA, what about it? So you know, we will talk a bit about data science and data. Data's all over. You have these big companies, Google and so on, producing an awful lot of data. We are producing that data for them. They use it. They say, thank you very much. You don't need to pay. But we do pay heavily with our identity, with all the ads they make out of this, and the special searches. And their ad money, they earn a fortune. But there is a lot of information. The estimate is that last year we had roughly 10 yottabyte. That is 10 times 10 to the power 24 bytes. An awful big number. Petabytes or petabytes and a few more. So we have lots of computers. And for example, we are doing sequencing, measuring DNA that produces a lot of data. And you know, there is a golden rule for computation or for computers, it's called Moore's law. Every 18 months, every 1.5 years, computer power doubles. Yeah? So you can calculate twice as fast, but you can also store twice as much stuff on all the disks that are there. Now, how many disks overall do you think to hold all of the information in the world? Will those disks, if you would buy them, could they fit into this room? Or do you more think about this whole building? Or do you think we need to cover Amsterdam with all of those disks? Or should Holland be wiped out, you know, with just a big pile of computer disks? Let's say it's this Dutch country. What's the purpose anyway? Let's take all those disks over it, you know, bury them once and for all, you know. I don't know. So, what do you think? Well, I don't know either. No idea, right? It depends on you. You should say something clever back. But sure, that depends on the technology. How large do you think that this should be? It's getting smaller and more powerful. Okay, I buy that. But then, there is a little molecule optimized in evolutionary times, and that has 3 letters. It's called DNA. And this is a fabulous molecule. All that information, I just mentioned, like 10 yottabytes, will fit on a few grams of DNA. And a few grams of DNA, you know, and really you have a bag like this, and that will contain all that information. So there is nothing like it. We as mankind have not come up with any technology, any way of storing information that gets any close to what DNA can do. And another beautiful point is, and here it comes, DNA is far more stable than a computer disk. These bytes will wipe out over time. You walk with a magnet, you know, put them on a disk, and already the disk can suffer from it, right?